Okay, so good morning, good day again to you guys. And so today we're going to take a look at your activity number two, your diffusion. Again, that is your main membrane physiology. And with that, we're going to take a look at the factors that affects diffusion, that affect diffusion. Okay, and so please note again that your plasma membrane or your cell membrane that is said to be selectively permeable. Okay, and so how is that so? It allows the water to freely pass through while regulating the movement of solutes accordingly. No? Okay, the intracellular fluid and extracellular fluid environment of the cells are aqueous solutions. Okay, and so you have your ECF and your ICF, your intracellular fluid or your cytosol, that is 90% water accordingly. And of course, the remaining 10% will be your dissolved substances. Okay. And so for this one, the differences in the concentration of the solvent and solute in and out of the cell determines the movement across the cell membrane. Okay. And so how is that? So of course, we need to maintain equilibrium between the inside and outside of the cell. Okay. And so we need to attain balance. Okay. And so that is for your, that is actually the use of diffusion. Okay. And so with that, in the laboratory, we did your uh, diffusion through agar gel using your stains methylene blue and methyl red. Okay, and so in this case, we were able, that is actually, we were able to demonstrate the effect of molecular weight. Okay, and so the effect of molecular weight. With that, if you computed the molecular weight of your methylene blue and methyl red, your methylene blue will have a greater molecular weight. Okay, and so for this one, that is why, based on that molecular weight, theoretically, it should give you what we call this, a smaller diameter. Okay, and so, or a slower rate of diffusion. Okay, and so with this, you are you are tasked to record your diffusion or the diameter that the stain has taken up, okay, in your agar plane. Okay, and so with this, your methylene blue, since it has a larger molecular weight, then again, theoretically, it should have a slower rate of diffusion, comparing it with methyl reds. Okay, and so again, I will tell you this one again, mas mataba, mas malaki, mas mas malaki, mas mabagal gumalaw. Okay, and so please remember that in the concept of diffusion in case of molecular weight. Ala, okay. Your diffusion here is defined as the movement of solute molecules from an area of higher concentration to an area of lower concentration. Okay, and so please note that these are your solute molecules. The solute molecules, they will move in case of diffusion. Okay, and so with this, your substances diffuse according to their concentration gradient. Okay, and so usually substances will move against a concentration gradient. Okay, and so for this one. And of course, that is to maintain equilibrium. Okay, and so to maintain equilibrium, if equilibrium has been reached, then there is no net movement. There is no net movement of your solutes in this case, thereby yes, in equilibrium. Okay, and so forth. This one. And yes, again, after a substance has diffused completely through the space, removing its concentration gradient or differences in the concentration, your molecules will have a net will have no net movement. Again, molecules will still move around in the space, but then there will be no net movement. Okay, and so with that, you already reach equilibrium. With this, let's take a look at the effects of your diffusion. There are actually several factors that affect the rate of diffusion of a solute. Okay, and so in this ones will include, you have here your mass of the solute. Okay, again, we did this one. And yes, you computed for the molecular weight and comparing it with the experiment that you did. Yes, your methylene blue, it has a smaller diameter, okay, in diffusion, okay, and so that again with this, the mass of the solute, heavier molecules move more slowly, okay, and so with this again, please remember, mas malaki, mas mabagal. Okay, and then you have your temperature, the effect of temperature for this one. In the experiment, we made use of cold and hot water. We made use of cold and hot water. Okay, and so for this one, as based on your end results, hot water, the diffusion of your food coloring in hot water is faster. Okay, and so with this, your temperature, higher temperatures increases the energy of the molecules, therefore increasing the rate of diffusion. In contrast, lower temperature, it will decrease the energy of the molecules accordingly, thereby decreasing the rate of diffusion on molecules. Okay, and so again, with this, heat will increase the energy of the molecules. And then your solvent density, we made use of dissolving sucrose into a water. And then after dissolving, you will let it stand and then added a drop of food coloring for this one. Okay, and so in solvent density, you have here as the density accordingly of the solvent increases, the rate of diffusion decreases. Okay, and so the rate of diffusion decreases. And yes, because movement will slow down when the medium gets denser. Again, the molecules will slow down because they have a more difficult time getting through a denser medium. Okay, comparing it with the beaker that contains only water, it has a faster diffusion rate. And then the distance that is traveled for your we did not actually do this, but then that is why we made use of it's the same size of beaker, a 50 ml beaker. Okay, and so in case of distance traveled, yes, it will what they call this the greater the distance that a substance that the solute must travel, the slower the rate of diffusion accordingly. And if you want to add here the size of the membrane accordingly, a larger membrane will favor the rate of diffusion. Okay, it will favor the rate of diffusion because there is a larger surface area for your solutes. Okay. Okay, moving on, let's take a look at your cell membrane, your lipid bilayer. Your lipid bilayer, this one is a part of your cell membrane. And yes, this is a two back-to-back lipid layer of lipids. Okay, and so you have your lipids present, you have your phospholipids, your cholesterol, and your glycolipids. And yes, you have here your phospholipids. It is the greatest among the three, followed by your 20% cholesterol and 5% glycolipids. Okay. And with this, your lipid bilayer, this is amphipathic. This is amphipathic. What does that mean? It has both a polar and non-polar part. Okay, and so for the polar part, you have here your phosphate head, and then the non-polar part, you have the fatty acid tail. Okay, and for your carbohydrate, I for your what we call this for your glycolipid, you have your carbohydrate group and your OH group that are present in them. Okay, and so giving you this, giving you a polar property also. Okay, and then yes, the presence of your membrane proteins in your lipid bilayer. You have the presence of your membrane proteins in your lipid bilayer. Okay, and so that is for your lipid bilayer. The concept of selective permeability, you have your plasma membranes that permit some substances to pass more readily than others. Okay, and so please note that the lipid bilayer or portion of the plasma membrane is highly permeable to nonpolar molecules like oxygen, carbon dioxide, and steroids, together with your vitamin, fat soluble vitamins. And yes, the lipid bilayer is moderately permeable to small uncharged polar molecules like water and urea. Again, please note that your urea, this is a waste product from the breakdown of amino acids. And your lipid bilayer, again, is impermeable to ions and large uncharged polar molecules like glucose. Okay, and so how will this substances pass through. Okay, and so you have if they are not allowed by your lipid bilayer. Okay, and so please note again you have the use of your membrane proteins present in the plasma membrane. Okay, your transmembrane proteins or your membrane proteins, they act as channels and carriers to increase the plasma membrane's permeability to a variety of ions and uncharged polar molecules. Okay, and so in the concept of this one, the more hydrophilic or lipid-soluble a substance is, the greater the membrane's permeability to that substance. Okay, and so going back to this one, sabi natin kanina, your steroids. Steroids, they are, what they call this, they are highly permeable. Okay, and so they can readily pass through the plasma membrane, the lipid bilayer. And please note that steroids, they are lipid soluble. Okay, and so comparing it with your charged molecule, uncharged polar molecules. Okay, but then with the help of your membrane proteins present, these substances that are blocked by your lipid bilayer, they can enter the cell. Let's take a look at your facilitated diffusion via carrier proteins. This one is a type of diffusion and a type of passive transport. Again, when we say passive transport, it does not require energy. Okay, and so the movement of solutes does not require energy for this. But then you need carrier proteins. Okay, your carrier, it needs carrier or transporter for this one. How is this so? Your solutes bind to a specific carrier protein from the other side of the membrane, let's say from the ECF, and then after that, your carrier will, what we call this, there will be a change in the shape of your carrier, allowing your solute to move. And after that, the carrier will discharge the solute. Okay, the carrier will discharge the solute at the other side of the membrane. After discharging, then the carrier will return to its original shape. Okay, and so and the rate is accordingly for your facilitated diffusion via carrier proteins, the rate is determined by the concentration gradient. Examples of substances that are transported via facilitated diffusion via carrier proteins, you have your glucose, your fructose, and your galactose. You have your facilitated diffusion via ion channels. Okay, your solute moves down its concentration gradient across the lipid bilayer through a membrane channel. Okay, and so you have your ion channels. They are integral transmembrane proteins that allow passage of small inorganic ions that are too hydrophilic to penetrate the nonpolar interior of the lipid bilayer. Okay, and so your ion channels allow passage of inorganic ions. In contrast to carrier proteins, there is no such thing as change in the shape because there is just an opening. The gates will open and then the gates will close for your ion channels. Okay, parts of the channel protein can act as gate and it will open and it will close. Okay, you have your ion channels for potassium chloride, and most numerous actually are ion channels for potassium. Okay, and the rest of your ions. Moving on, you have your primary active transport. It uses your active transport. They are, what do you call this? It utilizes energy that are obtained from hydrolysis of adenosine triphosphate. And yes, that is utilized. Your ATP is utilized to drive active transport. Without ATP, active transport will not take place. Okay. And so with this, it uses energy from hydrolysis of ATP, and energy changes the shape of a carrier protein to pump a substance across the cell membrane. Okay, and so please note, kanina, when we talk about your facilitated diffusion via carrier proteins, we did not talk about ATP. Okay, and so because again, that is a passive transport. It does not require energy. Unlike for primary active transport, it requires ATP. It requires energy in the form of ATP. In active transport, you have actually two types. You have the use of pumps and the use of vesicles. Okay? And so you have an example of your primary active transport. You have your sodium potassium pump. Okay, and so that is actually present in cells of the body. Okay, and yes, your application of your diffusion with this, of course, for us in the inside of the body, you have your continuous diffusion of your waste, like your carbon dioxide, and then diffusion also of oxygen. Okay, and so gas exchange in and out of the cells. Diffusion is very important in the body for the movement of oxygen from the lungs. Okay, and so to the tissues in carbon dioxide from the tissues and then transported to the blood and then finally exhaled in the lungs. Okay, and so movement of glucose also from the blood into the cell, and then absorption of nutrients, diffusion of nutrients into the cells from the gastrointestinal tract, and then detoxification of substances. After being detoxified, of course, they will be released outside of the cell for excretion. Okay, and so with that, let's take a look at your endocytosis. Okay, your endocytosis, this one is an example of an active transport. In your endocytosis, materials will move into a cell in a vesicle formed from the plasma membrane. Endocytosis. You have here an example of endocytosis. You have your phagocytosis. Phagocytosis is an example of endocytosis. You have your steps that will take place in endocytosis. Of course, the substance will bind first, okay, and so at the receptor. Okay, and so it will bind first at the receptor. And where can you find the receptor? They are present in the plasma membrane. Please note you have membrane proteins present in the plasma membrane, and one of those membrane proteins, they can function as receptors. Okay, and so they will first bind in your cell membrane and then followed by a vesicle formation. Okay, and so followed by a vesicle formation. After the formation of your vesicle, then they will be there will be uncoating, and then after that it will fuse with an endosome, and then recycling of receptors to the plasma membrane, and then finally degradation in the lysosomes. Okay, and so that is your basic steps in endocytosis. Okay, and then your exocytosis. During exocytosis, your membrane enclosed vesicles called secretory vesicles form inside the cell. Okay, and so with this, after they are formed, they will fuse with the plasma membrane, and then after that, they will release their contents into the extracellular fluid. This one is utilized for release of materials from a cell. That is either a waste product or a what they call this a substance needed for the ECF or for other parts of the body that will travel via the circulation. And then your transcytosis, this one is a movement of your substance into across and out of the cell. That is for your transcytosis. Okay?